Hello student, very good morning to all of you. How are you? I hope you all are happy, healthy and fine at home. Do you remember what were we discussing yesterday? I think we were discussed about the plus minus Subincanadine E. And how do we get? There are the two ways to obtain that Subincanadine E. One way from maleimide and second way from the acetoxysacinamide. By using these are the two precursor or the two starting material, they have derived that target molecule, and the speciality of this what we can call it as a synthesize that natural product in the laboratory, because it shows the cytotoxic biological activity, and therefore the structurally interesting and biologically important cytotoxic alkaloids Subincanadine A to Z were isolated in 2 to 14 milligram quantities from 100 gram bark of the Brazilian medicinal plant Aspidosperma Subincanum by Osaki and co-workers in 2002, that is represented in the figure number 1, and that how they are showing. Let us see here. See, we are going to find out the structures of, they have synthesized the Subincanadine A to Z, the A to Z, how that structures are there, that we are going to find out over here. So let's see. There is a general one structure which I am representing over here, this one, and then this structure further 5-membered ring. Here is a further 5-membered ring. This structure 5-membered ring, and here at this position there is a R group, and this nitrogen is a tertiary. Here attached, and this group is above the plane, and which is known as a R dash. Here are the methyl group. That methyl group also goes above the plane, and look at this nitrogen atom. Nitrogen atom has 4 valency, therefore positive charge comes on it, and here you can write as a, see that kind of the salt chloride ion, so that kind of the salt may be present over there. Now here there is a minus Subincanadine A, minus Subincanadine A, when there is R is equal to H, this one, this become as a benzene moiety, and R dash is equal to OH. Here if it is a OH, then that structure is a Subincanadine A. This is a structure of Subincanadine A, where this R is a hydrogen atom and this R dash is nothing but as a hydroxy group, that is a Subincanadine structure A. Now let's see now another structure, that is a Subincanadine structure C. Look at here, C. I am representing the letters. Here is a C. When R is equal to H and R dash also H, R dash also H, then that structure should be here R is equal to H, and here this R as well as R dash both are hydrogen atom, then that structure is called as a Subincanadine C. Now let's see another one that is a Subincanadine G, that is a Subincanadine Subincanadine G, when there is R is equal to hydroxy and R dash should be hydrogen atom, exactly opposite to the A, then it is known as Subincanadine Z. Subincanadine and the Subincanadine A are the alternative, where R is a OH and R dash should be H. Then next structure I am going to draw over here. So let's see another structure. What is that? Let's see properly. Again I am showing this moiety once again. Then there will be 6 membered ring like this, and then 5 membered ring once again, 5 membered ring, then this one again. Here is a what we can call it as hydroxy OH. Sorry, here remember one more thing we forgotten. Here should be double bond and this methyl group, and in the here also double bond and methyl group, positive charge Cl minus we can write over here, and after that here the methyl group. Look at, in this case methyl group is above the plane, but in the next structure exactly opposite that methyl group, you can clearly notice that methyl group is below the plane. In the first structure the methyl group is above the plane, and therefore we got as a 3 structures: Subincanadine A, Subincanadine C and Subincanadine Z, depending upon the nature of R and the R dash. There we got the 3 Subincanadines A, C and Z. Now this is also as a one of the structure, and this is nothing but as a structure minus or plus, here also as a minus Subincanadine B. Look at here, this is a structure of Subincanadine B. What the minor change over here? Here if you see over here this structure Subincanadine A, where the methyl group is above the plane, but in case of the Subincanadine B, where the methyl group is below the plane. So that is the minor change, but that minor change plays very important role in the biological activity, and that's why these minor changes becomes as a very big making as a very big changes or playing as a very big role in while exhibiting as a biological activity. Particularly these here, the one of the minus plus Subincanadine E showing cytotoxic biological activities, and therefore the scientists get attracted toward the synthesis of this natural product starting from various starting material they from the laboratory, and they can prepare that compound in a large scale on an industrial process, and that's there. Now let's see, these are the structure, 4 structure we have drawn here. So total structures are A to Z, Subincanadine A to Z. We have got so far A, then B, C. Now remains is D remains there. A, B, C, E, we are not getting over there. Only A, B, C, D, E, F, Z we got. So middle one, A, B, C and Z, 4 structure we got over here. One Subincanadine A, Subincanadine C and minus Subincanadine Z and minus Subincanadine B. These are the 4 structure we got over here, A, B, C and Z. Now remaining over here is D, E and F. These are the things, and this is our what we can call it as a main focus for the synthesis of Subincanadine E, and which is written, which is given as a heading over here, and that's why we are trying to synthesize that one. Let's see the remaining 3 structure. What would be the structure of these 3 that we are going to see over here. Now let's see the another structure over here. Another structure will be, now see here, this is the structure I am going to draw over here. This is a benzene moiety, and then there will be here hydrogen atom, and then we can draw the structure of this will be nitrogen, then this structure. Now there will be what we can call it as a, here should be double bond, here a double bond, and now after the double bond, and this should be below the plane. So 6 membered ring, here should be double bond, and here remains this moiety remains as it is. Indole moiety remains as it is. It is a chiral center, and this hydrogen atom should be above the plane. So these are the compound which we are going to discussing here right now. This is the structure where we will find here. Now look at here the structure which we are trying to get here. I am making some corrections over here. Observe those corrections, notice those corrections, and here one of the R group I am writing below the plane, that is R group over here. And now here is a R. Look at here, that is a minus Subincanadine D, when R is equal to OH. If R is equal to OH, that structure is called as Subincanadine D. And when there is a plus, now the optical rotation is changed over here. Plus this one is a Subincanadine E, when there is a R is equal to H. When R is equal to H, that structure is known as a Subincanadine E. So that is a structure we have drawn over here, and our main focus on that positive plus Subincanadine E, where R is equal to H. And finally, what will be the last structure? Let us see here. This is one of the indole moiety remains as it is over here. Here is the what we can call it as a, here the structure which we are going to show here is a nitrogen atom, then here will be 6 membered ring, here is a double bond, one methyl group over here, there is a carbonyl group, alpha beta unsaturated compound, and then this the bond which is going to attack over here, and this is a structure where there is a hydrogen atom that goes below the plane. Here also has one of the chiral center you can observe over there. And now here the double bond of this aromatic compound indole moiety remains as it is. This is one of the structure, and the name of this structure is a plus. Once again it is a plus this is a plus Subincanadine F. This is a structure of Subincanadine F, and this this scheme is represented as a figure 1. What is that? Potent cytotoxic alkaloids, Subincanadine A to Z. So that all the structure we have seen here, potent, these having as a potent cytotoxic alkaloids, and alkaloids, as we know that these behave like as a alkali, and therefore they are termed as a alkaloid. But alkaloids are basic in nature, similar to as a alkali metals, and if it can be taken in a small amount, they are very poisonous one. Alkaloids are very poisonous one, but if they are taken in a small amount, it can act as medicine. Remember here, and that's why these are very important role playing in a medicinal field, and therefore the approach to synthesize this kind of the molecule in the laboratory is highly essential. So plus Subincanadine E is also named as pericin. Is also named as a pericin, named as a pericin, and was first isolated from Picrolima nitida by Stockett and co-workers in 1982. Very at the beginning they have studied from this. Recently, Chem and co-worker proposed that the pericin is a common biogenetic precursor of two structurally unprecedented monoterpenoid indole alkaloids plus arboricidin and minus arboramine, figure number 2, which are shown in a figure number 2. Let's see what are those structure that I am going to, because we are discussing introduction of that Subincanadine. These all A, B, C, D, E, G, all the structures of the Subincanadine A to Z we have discussed, we have seen over here. Now the next, some of the structure which I am going to draw over here. Let's see. This is a structure which I am representing over here. Here is a nitrogen atom, double bond, and this moiety remains as it is. Then there will be a 6-membered ring. This is a 6-membered ring. Yes, this is a 6-membered ring. Now here one of the nitrogen atom over here shown like this, and this nitrogen further shown over here like this. There is a one hydrogen atom which comes out the plane. Now one of the nitrogen atom that attach over here, and now here is a methyl group which will be above the plane, and this is a structure. The name of this structure is a minus arboricidine. That is the structure of minus arboridicine, and the next structure is I am going to draw over here. All are having as a indole moiety over here. This one, not this one. Here is a double bond. Then there may be as a 6-membered ring. Then one more 6-membered ring attached over here, and there will be a 5-membered ring attached over here, and then here is a double bond. Here is a hydroxy group. It should be above the plane. At this junction there is a hydroxy group which above the plane. Here the hydrogen atom, that is a cis junction, and here is a double bond, and this is the structure. The name of this structure will be minus arbornamine. That is the name of that. And this is a figure number 2. Figure 2, and the title of that figure, biogenetic precursors plus Subincanadine E, pericin in bracket, pericin, derived novel, natural products is there. So plus Subincanadine E endures unique structural architecture, and in vitro and vivo you might be knowing. Vitro means reaction are carried out outside the lab, and vivo means reactions are carried out inside the lab. So that type of they are the biological word. Then plus Subincanadine E endures unique structural architecture and in vitro exhibits potent cytotoxicity against marine lymphoma, L-1210 cells, where IC15.3 microgram per ml, and human experiment epidermoid carcinoma KB cells, IC15 4.4 microgram per ml. A few new synthetic routes to the target compounds from figure 1 have been reported in recent literature. Zhai and co-worker in 2014 reported the first total synthesis of plus minus Subincanadine E, and development of new synthetic approaches for plus oblique minus Subincanadine E is essential from its exceptional structural feature, promising biological activity and establishment of stereochemistry point of view. Retrosynthetically, corresponding tryptamine derived maleimide would be the potential precursor for a total synthesis of plus minus Subincanadine E, which is represented in scheme number 1. Conceptually, the starting maleimide bears suitable functional groups for sequential 1,2 and 1,4 addition of Grignard's reagent, followed by intramolecular Pictet's Spenger cyclization to provide the desired tetracyclic hexahydroindolysinolone. Condensation of the above stated well-protected lactam with acetaldehyde to generate the exocyclic carbon-carbon double bond and essential functional group. Interconversion deliver the known advanced diol intermediate leading to the target compound. Moreover, R and S acetoxysacinamide may also serve as approximate starting material for enantioselective synthesis of plus minus Subincanadine E. In continuation of their studies on the use of cyclic anhydride to synthesize bioactive natural products, we herein report the synthesis of plus minus Subincanadine E and natural isomer plus Subincanadine E from the readily available corresponding imides as a starting material, which is represented in a scheme number 1 to 5, and that I think we will see in the next lecture. Till then, let us stop today here. Thank you. Have a nice day.